Over to Aaron, who's going to introduce today's student-invited speaker.
Hi everyone, it's my pleasure to introduce Zach Cheviron, who is joining us today for MVZ Lunch as one of our student-invited speakers for this semester. So every year the graduate students get together and nominate a couple of people to come, and Zach, I'd like to thank you for accepting our invitation. So Zach got his undergraduate degree at the University of Evansville and a master's at Illinois State. And he then went on to get a PhD at Louisiana State University in Rob Brumfield's lab in their ornithology museum before doing a postdoc at the University of California at Los Angeles, and then a second one at the University of Nebraska with Jay Storrs, which is where our professional lives sort of tangentially intersect, because Jay did his postdoc with Michael, who is my current advisor. We're all connected. And
That makes you like a first cousin.
yeah, yeah, and that's where Zach got started on some of the work that we're going to hear about today, and that's really been a major component of his research program. And that's adaptation to high altitude and hypoxia in deer mice in the Rocky Mountains in Colorado. And he's taken that work to the University of Illinois at Urbana-Champaign, where he began his career as a professor, and to his current position at the University of Montana in their biology department. In addition to high altitude and hypoxia, he has a bunch of other cool projects going on, including cold adaptation and work done with Fred Benham and others on salinity that you heard about at an MVZ lunch last year. And I think we're going to hear about high altitude today. So with that, Zach, thank you again for joining us, and I will turn it over to you.
Thanks, Aaron, for the invitation, and the grad students for the invitation to come. It's always super special to be a grad student nominated speaker, so I really appreciate it. This is actually my first time in Berkeley at the MVZ ever, without being a fan of this place for a long time, so it's really special. Thanks for having me. So today I'm going to tell you about work that we've been doing in my lab trying to understand the ways in which deer mice are adapted to high altitudes in the Rocky Mountains. So I'm going to spend a lot of time talking about how these small little mice live on mountaintops, and so you'll learn a lot about those specific physiology, but I think these sort of general questions are things that motivate a lot of us in this room, even if you're not into mountains. You have bad taste if you're not into mountains, but that's okay. These are things that are universal. So a lot of the work that we do in the lab is kind of motivated by a simple observation, and that is that colonization of novel environments often requires coordinated changes in independent but interacting physiological systems. So these are complex adaptations, regardless of how you want to try to define complexity. So there's a number of kind of subordinate questions that arise from this observation. One is, how are these changes coordinated both on physiological and evolutionary timescales? What are the limits of your ancestral physiology and your ancestral plasticity when you colonize these new environments? Does your ancestral physiology that often evolved in a different context, does that equip you to deal with new challenges? When may it not, and why is that? And what sets the limits on overall whole organism performance, and how can these limits be surmounted by adaptive evolution? I think many of us are interested in these kinds of questions. We tackle these questions in a variety of systems. So again, yeah, you know all about Fred's work in savanna sparrows, so I won't tell you about that today. We work in a variety of all vertebrates. Mostly, a lot of the work we do is focused on deer mice and high altitude adaptation. But if you're interested in any of this other work, I'm happy to talk about it later. And we focus on altitude for a variety of reasons. One is that when you're talking about sort of adaptation in these integrated systems, it's helpful to have a good understanding of the physiology that might be important in surmounting some of these challenges. So at high altitude, the challenges are obvious. It's hypoxic and it's cold. We know what the selective pressures are. We know how they change with elevation. We also have a good understanding of the physiological systems that should be associated with dealing with these challenges. And so these have to do with physiology that's related from everything from how individuals breathe to how they utilize oxygen in their tissues. And we'll walk through this physiology sort of over the course of the talk today. But this allows us to kind of root both predictions and then interpret the results of specific analyses because we have this foundation of physiology that was laid for us. So this thinking about high altitude adaptation as a kind of general framework for understanding how organisms deal with extreme environments has a long history, starting with some studies in the Alps. And I'm going to resist the urge to make a cheap joke about grad students carrying PIs in their field sites. Instead, I'm going to tell you about work that was done by Haldane. So this is Haldane's name that's probably familiar to many of you for Haldane's sieve or Haldane's rule. This is not that Haldane, JBS Haldane. This is his dad. So this is John Haldane, John S. Haldane, equally as much of a polymath as his son, and also an oddball in a lot of ways. But he was one of the first comparative physiologists. Many people refer to him as sort of the father of environmental physiology. And he led an expedition to Pikes Peak in the early 1900s that laid the foundation of what we think about as an acclimation response of lowlanders to high altitude. So this first expedition laid out the fact that red blood cell count increases as you go up in elevation. This is something we totally take for granted right now. This was discovered in 1911, really well documented by Mabel Fitzgerald, who worked in Haldane's lab in Oxford but couldn't attend Oxford. So she was not a student. She produced the first documentation of changes in hemoglobin concentration. That work is still cited today. She got a master's degree when she was 100 years old, 75 years later after this. So that was, Oxford said, fine, we'll give you a master's degree. Probably not up to her contribution. But this work has really laid sort of the foundation of what we think about as an acclimation response and other things you think about. And sort of has thought about the ways in which physiology sort of changes as a function of altitude. But some of this, the focus sort of changed right after Edmund Hillary and Tenzing Norgay summited Mount Everest in 1953. So this led to a revolution in sort of thinking about more about what limits performance at very extreme elevations. So this is work that was led by Griffith Pugh and Hillary to do a series of in situ physiological studies at very high elevations in the Himalayas. So this is the Silver Hut. This was built by Sherpas who carried this stuff up to this elevation. They built this hut and maintained it for 2 years. And this is some of the first trials of exercise performance at high altitude. They're just like really great contraptions, having people ride bikes at very high elevations. And this work laid the foundation of what we think of the constraints of high altitude. And a lot of this work culminated in this conclusion that at the summit of Mount Everest, you can barely support basal metabolic needs. So doing anything sort of athletic at high altitude was thought to be impossible. This was the physiologists of the day, sort of the leading opinion, which was upended by this guy. So this is Reinhold Messner, this grizzled old dude, and his buddy Peter Habler. So they were the first to summit Mount Everest without supplemental oxygen, right? So not just sitting up there maintaining life, but climbing mountains without supplemental oxygen. They did this in 1978, the first pair to do it. Reinhold went back in 1980, 2 years later, and did it solo, the first solo ascent without oxygen to the summit of Mount Everest. And so this caused a bit of a revolution among exercise physiologists, sort of thinking about what's humanly possible, right? Based on what we thought we knew about physiology. As of 2017, almost 200 people have done this. So it's far from being impossible. It's becoming, more or less common. And one of the latest people to do this is Kilian Jornet. Does anyone know who Kilian Jornet is? I think by any sort of objective measure, you could call him a badass. So his thing, he's a mountain runner, so he runs ultra marathons at very high elevations. He's a ski mountaineer. He has this project where he's trying to set speed records up tops of some of the most famous mountains in the world. In May of 2017, he ascended Mount Everest twice without oxygen in the span of 5 days, right? And so if you really want to get into really fine scale discussions about GPS tracks and how fast he was actually moving, you could go down a bunch of internet wormholes. It's not clear as to whether or not these are speed records, but suffice it to say, he's the first person to do this twice without supplemental oxygen. I bring all this up because we went over the course of, say, 50 years, we went from something that was seemingly impossible to be done physiologically to something like, nah, you can do it a couple times a week if you want to. It's really not that big of a deal. And so this sort of revolutionized the way we think about limits to performance. So right after these ascents that were thought to be physiologically impossible, people have started to think, okay, what is it about these people? There must be something special about them physiologically. This is one of the first efforts to try to figure out whether or not these elite mountaineers differ physiologically from other people. So what you're looking at here is data on VO2 max. This is exercise VO2 max if you run on a treadmill. Maximum amount of oxygen you can consume. It's a measure of aerobic performance. We'll be talking about it a lot today. Over age, and then a bunch of different groups. So these are elite marathoners, amateur marathoners, probably some amateur marathoners in the room, and sedentary controls. And these are the VO2 maxes of mountaineers, six mountaineers that climbed Everest without supplemental oxygen. So right here from this graph, they fall somewhere between amateur marathoners and elite marathoners. So not particularly remarkable here. This is where I don't mess with it. Right? So the guy to do it first is actually a little bit worse off than probably some amateur marathon runners here. And this is Kilian Jornet, who's a beast, right? So this is his VO2 max, one of the highest we've measured. So bizarre. There's good reason to suggest, well, maybe this isn't a very good measure for performance that's relevant for climbing mountains. So people have looked, this has been replicated too more recently. This is a paper from 2013. This is from the French National Alpine team. And their VO2 maxes, again, fall right in here with what are sort of amateur marathon runners. There are other metrics of performance. So this is just a measure of power. The point here is just that we see this graph's hard to make out, but we see no difference in power between elite mountaineers either. There's other sort of well-trained groups. And also, if you look at a variety of other physiological metrics, elite mountaineers don't differ from other well-trained groups in a variety of other traits. And I'm not going to belabor this, but there's nothing really that's standing out consistently that separates elite climbers from other groups of people that are in reasonably good shape. If you step back a little bit, though, and you expand sort of the evolutionary context in which we're asking these questions, then patterns begin to emerge. This is going to be probably a message that I don't have to give this group. This is a plug for comparative physiology. So as you expand the evolutionary timescale at which you're looking for these types of patterns, we can begin to see signals. And so some of the best examples have come from studies of indigenous human populations that have been at high altitude for varying degrees of time. And most of this is focused on indigenous Andeans and Tibetans from the Himalayas, and more recently, indigenous populations in the Ethiopian plateau. One of the things that's emerged from this work is that even though you can find signatures of physiological adaptation at high altitude, there's very little convergence at the level of phenotype. So each of these populations seem to solve this problem in different ways, right? So not a clear signal that's emerging from a lot of this work in what the convergent phenotypes might be. This has been best characterized in Sherpas, right? So Sherpas are famous for doing things like this, right? So these are the guys that carry the stuff that allow rich people to climb Mount Everest, right? So they set up the infrastructure to do that, and because they're able to do these things, they've been subject to a variety of physiological studies, and there's a whole host of traits that separate Sherpas from people of European descent. And if you're interested in any of this, I'll tell you more about it later, but all I'm doing here is just showing that we do see physiological signatures that separate Sherpas from other kind of human populations. And of course, most recently, this has been subjected to studies of the genetic basis of some of these differences. So these are genome scans or GWAS types of analyses that are looking for loci that have the signatures of selection or that are associated with some of these phenotypic traits. And again, we don't see a ton of convergence. So the way that this is color-coded is, but the red would be loci that are outliers in Andeans, blue in Tibetans, purple for Ethiopians, and this is the union of those two. But you can see that there's a variety of loci that are targets of selection in different indigenous human populations. So although we can detect signatures of selection, there's not really strong, consistent patterns in human populations as far as we know right now. If you step even further out and start to ask, okay, well, it's not just humans that are dealing with these situations. There are many animal groups that are specialists at high altitude. So one of the first to be studied is bar-headed geese. So as Reinhold is struggling up the mountaintop, taking 15 steps or so every 5 minutes, there are birds that are flying above him, right? And so one of these is bar-headed geese. Bar-headed geese have been the subject of a variety of physiological studies, and they differ from other lowland birds in virtually all aspects of the oxygen transport cascade. So they're famous for having hemoglobins that have a high O2 affinity. They differ in the size and the efficiency of their lungs. They differ in the capillarity of their muscles and the function of their mitochondria that are in those muscles. So a variety of wholesale changes to their physiology to allow that kind of high altitude flight. But even after the Pikes Peak expeditions and what we know sort of from other comparative groups, there's a variety of questions that we don't really know the answer to yet. We don't have a really good handle on the physiological traits that should best predict aerobic performance at high elevation. We don't know whether or not acclimation responses, so kind of ancestral plasticity, whether or not that mimics evolutionary adaptation. So things that are plastic, do they change in the same direction that evolution favors at high altitude? And of course, we don't know the genetic basis of these traits that confer these physiological benefits at high altitude in most cases. And that's because there are either sort of experimental logistical challenges associated with working on humans or bar-headed geese, or other kinds of genetic components or genetic infrastructure constraints for working on things like bar-headed geese or things that are less genetic models. So we've been trying to find a sweet spot where we are working on a rodent model that is adapted to high altitude, that's amenable to experimental and genetic manipulation, and is supported by a variety of sort of genomic tools, and we think it sort of fills this niche pretty nicely. So this is the deer mouse. This is one of the most widespread North American mammals. It has the broadest elevational distribution of any North American mammal. It's found on the floor of Death Valley all the way to the summits of the highest peaks out west, and so that allows us to make comparisons of populations that are native to different elevations. And deer mice have been a model for high altitude adaptation for a long time, so they're famous, at least in some circles, for hemoglobin polymorphisms, and some people in this room have worked on this question. They're famous for these hemoglobin polymorphisms that segregate with elevation, so high altitude deer mice, much like bar-headed geese, have a hemoglobin variant that has a very high oxygen affinity. And that's replaced by a low oxygen affinity at low altitude. So this was first described by Lee Snyder in the 70s and then was sort of fleshed out by my postdoc advisor, Ed Michael, in the late 2000s. This work continues on to today. We know that this variation in hemoglobin function is associated with variation in VO2 max. So this is the same VO2 max measure that I described before, but in this case it is cold-induced. And everything that I'll be talking about from here on out is also a cold-induced VO2 max. So rather than running mice on treadmills, which we have done, we put them in cold boxes and then we make them shiver to their maximum intensity to maintain a constant body temperature. We think this is an ecologically relevant measure. And so you can think about this cold-induced VO2 max the same way you think about an exercise VO2 max. And we know that individuals that have a high-altitude hemoglobin genotype have higher VO2 max under hypoxia than their counterparts that are
How tightly correlated are those two measures?
VO2 max and
running versus cold.
oh, so running is higher, and it is in most vertebrates, but there is a tight correlation. So they're related to one another, and the physiological systems that underlie them are similar, at least from the cardiorespiratory kind of perspective, not the locomotor muscles. But so you can think of them as one proxy for another. But I'm going to be speaking specifically about cold-induced VO2 max. We also know that variation in this cold-induced VO2 max affects survivorship in the wild. So this is a mark recapture study that was done by Jack Hayes and Candace O'Connor that just showed that individuals that have higher VO2 max, cold-induced VO2 max, are more likely to be recaptured over the course of a season than those that have low VO2 max. So this allows us then to tie variation, at least in some genotypes, to variation in whole organism performance that affects survivorship in the wild, and presumably fitness in the wild too. We also, so I think an important thing to kind of keep in perspective is that this variation, so hemoglobin variation and its functional effects on physiology is embedded within this cascade and falls sort of in the middle of this pathway. And so when I started in Jay's lab and sort of continued in my lab going forward, we've just been asking questions. Do we see changes in other steps in this pathway as well that would complement this variation in hemoglobin O2 binding affinity? And it should exist based on biochemical trade-offs that are associated with having a high oxygen affinity. But it wasn't known. And so we've started just to kind of flesh out what other parts of this pathway do we see differences in Highlanders too. So we do this by taking a kind of a complementary kind of field and lab perspective. So we measure a variety of physiological traits in the field. This allows us to have a snapshot of what the traits look like in the wild. This is a combination of both genetic and environmental factors, but this is what the phenotype looks like in the wild. And then we have breeding colonies of mice that are derived from different high and low altitude populations that we can acclimate to different hypoxia regimes. So these are hyperbaric chambers that we built. This allows us to simulate elevation basically just by pumping air out of them and reducing the pressure inside. And these are inside of cold rooms, so we can manipulate temperature and pressure at the same time. So then that allows us then to separate those genetic and environmental components on all this physiology that we'll be dealing with. So one of the first attempts to do that was just to ask, okay, so how do we expect VO2 max to vary with respect to acclimatization history? So what I'm showing you here is, these are the measures of mice from the summit of Mount Evans, which is one of the two 14ers in Colorado you can drive to the top of. Pikes Peak is the other. And then mice from Lincoln, Nebraska. So they'll be our highland and lowland comparisons for the Great Plains. And these are measures on the day that we catch them in this VO2 max measure. We also take mice from the field and bring them into captivity and deacclimate them to a common garden for 6 weeks. That's what this is. And then we breed and test their offspring. So this is F1. We've carried this out several generations at this point. And that's what's looking at this time point here. You can see that in all time points, the Highlanders outperform the Lowlanders, but there is an environmental component to it too. And so a lot of what the work that we're trying to do is to suss out these genetic differences versus the environmental influences on this whole organism performance. So the kind of small questions we'll focus on for the next few slides is, one, do Highlanders and Lowlanders differ in other steps of the oxygen transport cascade to complement those hemoglobin differences we know exist? And if they do, which of these traits is the best predictor of aerobic performance at high altitude? We'll do this by taking kind of a modeling approach that is rooted within this oxygen transport cascade. So we can represent this oxygen transport cascade in a variety of, in a modeling framework that's basically trying to follow variation of or movement of oxygen across tissues through the circulation and into diffusing into muscles and how it's utilized. So all of these steps can be represented by a series of equations that are not necessarily important, but they at least for our purposes in this talk, not our purposes in doing the work, but for the purposes of this talk, these are things related to how well oxygen diffuses across the lung-blood barrier. So this equation here is basically a diffusion equation that's modeling the movement of oxygen from the lungs into the blood. At each of these steps, we can parameterize these equations with physiological data that we collect on individual mice to model these steps of the oxygen transport cascade. The reason that we want to do that is once we have a model parameterized, we can turn knobs to calculate or to change in silico, to change different aspects of the underlying physiology. So for example, you can increase breath rate by 20% and leave all the other parameters in the model the same, and this allows you to calculate the proportional increase in oxygen consumption. Right? So if you increase breathing by 20%, the proportion, if that VO2 goes up 20%, then it has what's said to be a control coefficient of 1. So this, just changing this single variable explains all the change in the whole organism performance. If you change it by 20%, it has no effect. It has a control coefficient of 0, right? So these range from 0 to 1, and real physiological traits are somewhere in between there. But what this does is it allows you to place importance on specific steps in a modeling framework which ones we would expect to have the greatest effect on overall organismal performance. So if you do this modeling exercise in humans, this is what you get. So the first thing I want you to take from this is that control is not equally distributed across all steps. In fact, things that are related to blood O2 carrying capacity are actually quite low compared to things that are related to oxygen diffusion. So this is diffusion. Each of these are color-coded by each of these steps here. What's most important for humans is the step that is involved in diffusion of oxygen from the blood into the tissues. So we've been working to parameterize these models, and by we I mean Kevin Tate. So Kevin Tate, in order to do this, we need to draw venous and arterial blood samples while the mouse is shivering at its maximum intent. So we, the royal we, catheterize mice and then are able to make these blood draws at specific times during these cold stress trials. And by measuring variation in blood gases from those blood samples and other aspects related to heart rate and things we know about the blood oxygen carrying capacity, we can parameterize these models for deer mice, and we've done it, and this is what happens. So this is not particularly interesting probably to this crowd, but there are some things that I want to point out. One is that, again, we see the greatest control vested in this diffusive step. Control is not equally distributed again. And so again, these lower level steps are the most important in driving aerobic performance. And just file that away. This has also been done in bar-headed geese. And again, you see even more control vested in these diffusive steps. There's physiological reasons why this would be the case that if you're interested, I can tell you about that. But it has to do with the overall efficiency of the avian respiratory system. But for bar-headed geese, 60% of variation in VO2 max is vested in this diffusive step, right? So this is by far the most important step in determining VO2 max. So this is interesting physiology, but probably not what we're really interested in here. So in my view, the really kind of interesting question is whether or not we can use these control coefficients to make predictions about evolutionary divergence. Do the most important traits diverge the most between highland and lowland mice? If they do, this is what you might expect. You might expect greater phenotypic divergence as a function of control coefficient. So we asked that question basically by making comparisons of mice that were born and reared in our breeding colony. So these are mice that have been in the colony for more than 2 generations, and we're looking at just standing variation in these traits, or constitutive differences in these traits. We don't acclimate them to hypoxia. This is just the genetic component of variation and different physiological traits that impinge on each of these steps. So we measured a bunch of stuff, and they're color-coded by the step that they're relevant to. So we measured things like breath rate, when they're exposed to an acute hypoxia challenge, their tidal volume. We measure the oxygen saturation of their arterial blood, things related to their blood oxygen carrying capacity, and things related to these diffusive steps. And then what we did was we just made comparisons of means between the highlanders and the lowlanders. And we expressed those as Cohen's D. This is just an effect size based on a t-test so it's the effect of being a highland versus a lowland mouse. And what you see is an overall positive relationship here. So the things that have the highest control coefficient, things related to diffusion phenotypes, show the greatest divergence between highland and lowland deer mice. And things that have very little control, make little contribution to aerobic performance, have relatively low divergence compared to others. So I don't think this has been shown before, and it's nice and it's intuitively pleasing. I like this result. The most important things diverge the most, right? And having these control coefficients allowed us to kind of think about it this way. Some of these traits, what they are, is their capillarity. We've known for a long time that the highlanders have more blood vessels in their skeletal muscle than do the lowlanders. This is a representative histological image. They also differ in their fiber composition, so more aerobic fibers than glycolytic fibers. These are differences that we know are genetic, and they differ between the highland and lowland mice. So the next question then is whether or not these acclimation responses kind of mimic that pattern of evolutionary adaptation, right? So the pattern of evolutionary adaptation is that divergence is greatest for the traits with the highest control. So is the acclimation response, does it also show that pattern? So do we see greatest acclimation as a function of control coefficient, right? So if this were the case, this would suggest that ancestral plasticity is adaptive and is moving in the same direction as favored by natural selection. So to do this, we took advantage of the same lines of mice. We did these experiments In the lab in our hyperbaric chambers, we treated individuals from each population with either a normoxic control or hypoxia. This recapitulates this difference in VO2 max, and then we asked how the differences in trait values varied within Highlanders in each of the two treatments and within Lowlanders in each of the two treatments. We asked questions about the acclimatization response in both of those populations. So same traits, and what you can see here is a different pattern. So your eye might see a negative association here, but it's not significant. What is most important is that the highest control, the things that are most important for determining VO2 max, don't acclimate, at least on the time scales of our experiments. So they have very low effect of this treatment in both Lowlanders and Highlanders. Okay? So that can be kind of counterintuitive for people to think about, so I just kind of want to show you what these data look like, right? So for these high control traits, what we see is a strong population difference, but very little acclimation, right? So the traits that are most important, they don't respond to this hypoxia treatment. Other traits do, so in this case, these are things related to breathing. You can see in the Lowlanders, they acclimate quite well, but in the Highlanders they don't, and so this is expressed in this way. This is tidal volume, where the Highlanders have an overall high value here, and the Lowlanders achieve that phenotype. So this is a significant population by treatment interaction. We also see situations like this, where the acclimation response of the Lowlanders overshoots the trait value that we see for the Highlanders. So this is hematocrit. It's the proportion of red blood cells. It's the thing that most people think about when they go to high altitude. In this case, the Highlanders are the Lowlanders are super plastic, but the Highlanders less so. And we think there's good reason for why that would be the case. So many of you are some of you are at least familiar with some of this story. So hematocrit is the proportion of red blood cells. When you go to high altitude, you make more red blood cells. That's a good short-term strategy. It does allow you to carry more oxygen through your blood, but it has a downside, and then it makes your blood thick. So when you're exposed to chronic hypoxia, that can actually be counterproductive for oxygen transport to the tissues. And so we see a blunted erythropoietic response in the Highlanders compared to what the ancestral condition. This is not just restricted to highland and lowland deer mice, so these are two other species, Paramiscus polynotus and Leucopus. These are strictly low elevation species. They all show the same kind of convergence on that high hematocrit, so the Highlanders seem to be unique there. This is exactly what we see in other highland specialists, so human Tibetans are famous for this. So human Tibetans that are living on the Tibetan plateau will have a red blood cell count that's similar to mine living in Missoula, which is like 3,000 feet, right? So not a sea level value, but much lower than what they should have sort of at that elevation. And of course, this elevated hematocrit over lifetimes is associated with a specific disease called chronic mountain sickness. So indigenous Andeans have very high hematocrits over the course of their lifetime, and that's associated with damage to the microcirculatory system, overloading the right heart, a number of problems. So, just to summarize what I've shown you so far, population divergence is greatest for the traits with the highest predicted control over whole organism performance. Again, I don't think this has been shown before. The lowland mice exhibit poor acclimation capacity for the traits that are most important, and this may mechanistically limit their acclimation capacity and their ability to achieve a high VO2 max after hypoxia exposure. And maladaptive side effects may outweigh marginal benefits in acclimatization responses with low control traits. And again, knowing what the control coefficient is allows us to make more quantitative assessment of these side effects. It allows us to contextualize it. Okay. So the summary so far is I started out by by telling you we were interested in whether or not other steps in the O2 transport cascade are modified between Highlanders and Lowlanders. This is what we know so far. Basically, every step has been modified. We've published this in a variety of places. But every step in the O2 transport cascade has been modified in the Highlanders to achieve this higher level of aerobic performance. And those traits differ in their degree of plasticity. So what I want to do now for the last little bit is just to think about what are the genetic basis of some of these traits at high elevation. So we're starting to make some progress here too. We're taking two approaches. One is kind of a classical QTL map, and we're making crosses and doing that. I'm not going to tell you about any of that work. Instead, I'm going to tell you about work that we're doing kind of a population genomic framework, and this is work that's being led by two postdocs in my lab, Rainer Schweitzer and John Vallada. So the data set I'll tell you about now is one that many of us in this room are using. So this is an exon capture. So we designed a custom exon capture array for deer mice. And I'm going to show you data that's going to, again, compare these two focal populations, but we're going to expand the geographic sampling for some of this population genomic work. And first, it's going to include mice from Merced County that came from this collection here. Michael lent us those data. So this allows us then to polarize changes in a single high elevation population. Now we have two lowland populations and a single high elevation population for this first set of analyses I'm going to tell you about. So the first thing I want to show you is just overall structure among these populations. These are just pairwise FSTs for all three populations for a bunch of LD prune SNPs. And you can see that overall it's pretty low, right? And it's not strikingly different really between the three populations. They're all more or less equally divergent from one another. You can visualize this sort of in a PCA plot. So this is just the PCA plot that separates out the three populations. So some structure. You can do an admixture or a structure plot or something like this. So all three populations are genetically distinguishable from one another with these data. So what we wanted to do is first to kind of scan, just do a quick scan for signatures, loci that may show signatures of selection. We started with this population branch statistic, which was developed in a study of Tibetan Highlanders. All this is a polarized FST comparison. So we have 3 FST measures and all pairwise comparisons, and we're looking for situations where FST is much greater for Highland comparisons between the Highland compared to the other 2 populations, so much greater than the overall kind of background structure. And what this tells us is that tells us loci that have extreme allele frequency differences in the Highland population relative to the other 2 elevation populations. So we generated a distribution of these PPS values. This is an underwhelming plot, but this is what it looks like. So then we calculated a null distribution of what these PPS values should be based on a demographic model from the same data. I'm not going to show you the model, but if you're interested, we can talk about it. So what you're looking at here in the green line is the mean of this distribution. The red is the cutoff for the 99.9% of the simulated distribution. This is the top 0.1%, anything beyond here. And again, that's a simulated distribution, not an empirical one. And so if you ask what's sort of over this, there's almost 2,200 genes that fall outside of this. That's a lot of genes, and it's kind of hard to think about. So we thought about ways in which we could pare this down a little bit. The next analysis we did is we just wanted to know, okay, of these PBS outliers, how many of them are strongly correlated with elevation if we were to expand the geographic sampling here? So these are sampling localities throughout the Front Range of Colorado, a few more Great Plains populations. The data set has 160 mice. We pruned that data set for unrelated mice based on relatedness score. And then we did this RDA analysis, which is basically just asking about genotype to environment association. There's a bunch of ways to do this, but we used this RDA analysis, and all we're doing is looking for loci that are showing an association between elevation and genotype. So when we do that, we ask what the overlap of this is. We've cut this gene set quite a lot, cut it in half. So there's many genes that show this elevational association, but a subset of those are selection outliers from the PBS scan. We performed one more analysis where we calculated delta pi, so this is the difference in pi for the high elevation mice. So the idea here is we're looking for loci that show low levels of diversity in the Highland population. This could be signatures of recent sweeps at high elevation. And then the union of these gets us down to a data set that's manageable, say 350 genes. We've done gene ontology analysis on this, because you do that. I'm not gonna tell you about that. I think there's more direct ways to kind of start to think about what those do. But one thing I can tell you is that there's candidates in that list that impinge on all steps of the oxygen transport cascade. So I'm just going to show you some exemplars here. We know that Highland and low altitude mice differ in their ability to transport oxygen across the lung into the blood. These are 2 genes that are involved in regulating blood pressure. So blood pressure and vasodilation is important in that process. So these are interesting. We also know that they differ in their erythropoietic response, so I've shown you that already. This is ceruloplasmin. It plays a key role in red cell development. We see a strong clinal variation. And ceruloplasmin is an outlier for all three of our selection tests. We know that the Highlanders and Lowlanders differ in their muscle phenotypes, and these genes have been implicated in relating to these physiological or these phenotypic differences that influence diffusion capacity. Again, remember those are high control traits. And we also know that the Highlanders and Lowlanders differ in their overall aerobic metabolism pathways, and these are core metabolic genes. So these are just associations that are interesting. I want to tell you about some work that we've done to kind of try to associate one of these loci with phenotypes in the mice. So this is EPAS1. Many of you probably know about EPAS1. This has been a target of selection in Tibetans and many other things that live at high altitude. It's interesting because it encodes a transcription factor, HIF2, that coordinates a whole bunch of physiological responses to hypoxia. So many of the things that we're interested in, things related to vascular growth and fiber composition, again, high control sorts of traits. It's related to erythropoiesis. That's the connection in high altitude Tibetans. It also influences heart rate and breath rate through its influence on the secretion of catecholamines. And we see a strong clinal signature in EPAS1. It's an outlier in two of our selection schemes. Again, this has been shown to be a target of selection also in high altitude Tibetans, and then a number of other things that live at high altitude. So first I'll show you that the EPAS1 allele frequency varies with elevation. So this, what you're looking at here is a SNP genotype between high and low altitude populations. We've been focused on the front range up until this point, but we've also included samples. Again, here we see the same variant at high elevation in the Sierras, for instance. So it's not something weird about Lincoln and Mount Evans. There seems to be a consistent pattern with elevation. One thing about trying to tease out the effects of EPAS1 when you're looking at individual mice, or from our two lines, our Lincoln and Mount Evans line, is that it's confounded by population structure. So we've generated a bunch of crosses to break that up, but that takes time and we're somewhat impatient. So we took advantage of the fact that this locus is polymorphic on the summit of Mount Evans. So both alleles segregate in that population. And that means you can go to that population, you can find individuals of all three genotypes on an otherwise totally admixed genetic background. So we developed a way to genotype individuals in the field, just with the restriction digest. This is Raina doing it. She's very excited about it. Very excited. This is actually one of my most satisfying scientific achievements. It's just so nice to genotype a mouse in the field and then do some physiology on it. So we were able to do that, and then we measured genotypic responses to acute hypoxia challenges. So first we did an acute exposure, hypoxia exposure, and then we took them even deeper into hypoxia to measure the transcriptional response of HIF target genes, right? So this is a transcription factor that may influence the expression of many target genes. And this work was led by, the physiology work was led by Katie Ivey and Graham Scott, our collaborators at McMaster University. So we measured a variety of things that HIF could impinge on. So first we used plethysmography. So this allows you to measure breath rate and tidal volume in the field. And so we didn't see any difference in any of these breathing phenotypes by genotype. So no effect on breathing for this genotype. We did, however, find a difference in heart rate. So this is a pulse oximetry collar. So this is, you've probably had these put on your finger at a doctor's office. I'll measure your pulse rate by putting a little sensor on your finger. This is the exact same technology, but it's a collar. We have to nair the necks of the mice, though, to get the hair out of the way. And then once it's gone, you put these collars on. You can measure heart rate. You can also measure arterial saturation with these things. The Highland homozygotes were better able to maintain heart rate at levels of hypoxia compared to the two genotypes. And so what we're simulating here, this is sea level conditions, this is Mount Evans conditions, and this is even higher. And the greatest difference occurs at this ecologically relevant simulated elevation. So a strong effect on heart rate. We also saw some trends towards increased capillarity in the individuals that are carrying high altitude alleles, but this is not a significant difference, but a trend in that direction. And then finally, we wanted to know whether or not we see any genotypic effects on gene expression, again, because this is a transcription factor, we might expect it. Because we saw a difference in heart rate, we decided to focus on the adrenal glands. So again, HIF is known to regulate heart rate through the secretion of catecholamines. These are often called sort of fight or flight hormones. So the adrenals secrete catecholamines, things like adrenaline, and that can affect both blood pressure and heart rate. So we focused on those tissues. And what I'm showing you here is just sort of an overall signature of variation in the expression of these genes. So the gray distribution is genes involved in catecholamine biosynthesis and secretion. And the gray is just a randomly chosen subset of other genes that are not involved in those pathways. And what you're looking at here is the fold change difference between reciprocal homozygotes, so individuals that are homozygous for the low altitude allele, those are homozygous for the high altitude allele. And what you see is a shift, a more negative shift in individuals that are carrying the high altitude allele. So overall, in this overall pathway, we see a downregulation of catecholamine-related genes in individuals that are homozygous for the high altitude allele. The same pattern emerges if you compare heterozygotes to lowland homozygotes. That's the overall global pattern. There's a number of genes that are interesting here. Some of these you've already heard about. I already told you about ceruloplasmin. So these differ in expression by genotype. Angiotensin's involved in blood pressure regulation. These genes are also outliers in our selection scan. So this is the clinal variation in ceruloplasmin. So we see a difference in gene regulation among EPAS1 genotypes. And here the gene is not the exact same, but they're involved in the same family. Both of these are angiotensins. So just to conclude this part of the talk, we see many hypoxia-related genes exhibit patterns of extreme allele frequency variation at high altitude. One of these is EPAS1. It's a transcription factor that's been shown to be under selection in a variety of other Highland specialists. But in other Highland specialists, we don't really know the phenotypic effects of this variation. And in the one case that we do, humans, it's associated with red blood cell count. We don't see that in mice. Instead, we see an association with heart rate. Again, this is highly pleiotropic and maybe not surprising, but that's the result that we have. So the homozygotes are better able to maintain their heart rate under hypoxia, and genotype effects.
For your exome capture data, you collected some promoter regions as well as
We did, yeah.
If you look at your SNPs, do you see enrichment for coding versus non?
So, okay. So, yeah, I didn't actually even describe the mutation of the EPAS1 mutation I'm talking about either very well. But okay, so the promoter regions, all we did was we said, okay, give us 2,000 flanking bases outside of the coding sequence. So, hoping that there's some promoter regions in there. One thing that we do with these Go enrichments, and I kind of was a little flippant about it, we do see enrichment for hypoxia response elements. So those are the binding sites for various HIFs. There's 3 HIFs. And so we do see an enrichment for those binding sites among outliers. Yeah, but don't know anything beyond that. And so, but your question was about the distribution of coding versus non-coding. I don't know the answer to that, like how many outliers are coding versus non. I think mostly non, but I don't know the exact proportion. Yeah.
So this is early in the talk. Why do you think that mice and birds are so much better at the oxygen tissue diffusion than humans? That was the one where you had like 60% of tissue diffusion you get in birds.
Yeah. So for birds, their respiratory system is totally different than ours and is much more efficient in a lot of ways. So what that means is that oxygen saturation in the arterial blood, so after it leaves the lungs, is super high regardless of the elevation they're at. And so because of that, it can't contribute very much at all, right? And so that's why, because all this is a proportion, it all adds to 1, that's why it's so large for bar-headed geese, because that's where you can get some variation in that. So some of that is a mathematical function. But why is it, so those are lowland, those are just humans, right, that are, not trained or anything. And so they're not adapted to altitude in any way that we might think that. And so you can think of some of this as kind of an evolutionary progression. It kind of makes sense that selection may favor the highest control traits. And if you don't have a history of selection, then you wouldn't get convergence and investment in those steps. Does that make sense?
Yeah.
Yeah.
I have a question about your control stuff.
Yeah.
So when you are comparing the plasticity in the hypoxic conditions, you're assuming that the control coefficients are the same as under control environments. Is that the case? Because I know in terms of metabolic control, if you look at even just in a mitochondrial electron chain transport chain, control during a state a high ATP producing state versus in where electrons get held up versus in a resting state where it's leaking are very, very different.
Awesome.
And so have you accounted for that?
So I wasn't clear. So great, really good question. So the control coefficients that I showed you are assuming hypoxia. So it's under a hypoxic scenario. If you do the same calculation under normoxia, they change.
Yeah.
And the reason for that is because at high altitude, the premium is put on loading oxygen in the first place into the blood. That's why a high oxygen affinity hemoglobin is good.
Yeah.
But at low elevations, that's not the case. Oxygen is saturated, much like for the bar-headed geese I was just talking about. So at low elevation, our arterial blood is almost entirely saturated as we're sitting here right now. And so if you have a high oxygen affinity hemoglobin, that's actually bad, because then it's just bad at delivery.
Yeah.
So those control coefficients do change depending on whether or not you're talking about normoxia or hypoxia.
Right.
The numbers I showed you were for a hypoxic condition. Does that make sense?
Yeah, that makes sense. But I guess why wouldn't you use the matching control coefficients for your that? So for the control when you're looking at degree of plasticity.
Yeah. Because we're acclimating to the hypoxia. And so I think about the way I was thinking about it is that we're trying to explain traits that contribute to aerobic performance under hypoxia.
Okay.
And the acclimation is from normoxia to hypoxia for both. And so those seem to be the most relevant, at least in my mind. But I'd love to argue about it. Did you have a question?
Yeah, I was struck in the graph of allele frequencies for EPAS1 going from high elevation to low. So you have at high elevation, the high elevation allele is a very high frequency, and the converse is true at low elevation. Like there are no high elevation alleles at low. But there aren't a lot of intermediate frequencies going up and down there. So I have two questions. One is, what do you know about the cost of the high elevation allele at low elevation? And the second is, how much gene flow is there?
No, so we don't know for EPAS1, but that Klein Center is in almost the exact same place that we see for the hemoglobin genotypes, and there the costs are well described, right? So it's basically this, at low elevation, a high oxygen affinity hemoglobin is bad at delivery, so it's selected against. That's why it doesn't just spread up and down the mountain. Why it's so sharp is not totally clear, but it probably has to do with population structure, right? So, the challenge of hypoxia is basically linear, so you might expect kind of a linear, you don't see that at all for anything. We don't have a good understanding of where, we know that Lincoln and Mount Evans are genetically distinct, sort of in a structure kind of framework, but we don't have a good idea where background kind of population structure is. If you're going to make Kleins for just a random locus across the genome, we don't know exactly where that Klein Center is, but I would bet anything that it's basically in the same spot as all those. So I think that's a signature of just population structure, maybe secondary contact, I'm not sure. Do you have ideas?
Well, I was just wondering if there are places like in the White Mountains where, Lee Snyder, I think, worked in the White Mountains, where you can get a sharper elevational change over a shorter geographic distance, and whether that might be informative.
Yeah, so that the EPAS, well, so both the globin genotypes and the EPAS1 genotypes also occur in the White Mountains too. One thing we don't have a good feeling for is how many independent high elevation colonizations there are, even. We don't even know the answer to that. Which I always think we need to know the answer, but we haven't done anything about it. So yeah, it's a good idea.
I can have a related question, which is that do you think that the fact that you have this Klein, that this one species is distributed from lowlands to highlands, influences the targets of selection relative to a different species that's just confined to high elevation?
For sure. Yeah. So there's definitely, we know there's gene flow, they're distinct. So yeah, they have to deal with these trade-offs, whereas a specialist doesn't. And I mean, some of the things that you see sort of we just don't have that many comparative models, but if you just ask, so in things that are definitely independent high altitude colonizations, sometimes we see convergence at these physiological traits, but for example, sometimes they're different in things that are specialists versus generalists, like maniculatus is. So an example of that is like the proportion of carbohydrates and fats you use to power VO2 max. So phylotus mice in the Andes are basically elevational replacements of one another. And they show a different metabolic phenotype than do these generalists. The maniculates basically show an opposite metabolic fuel use pattern. And so that might be related to the fact that they don't have these trade-offs of trying to optimize, what's beneficial across all elevations, more of a specialization kind of thing. But I don't think that we have enough data to really know what those traits are. Yeah.
You mentioned at the beginning of your talk that there were a lot of coordinated adaptations that take place during high altitude adaptation. What about temperature regulation relative to oxygen consumption? Any data on that yet?
So the question, so you're just asking whether or not temperature compounds the effects of hypoxia?
Or if there's any relationship between temperature regulation and oxygen consumption
Oh, yeah.
in these animals?
So there's definitely a relationship between oxygen consumption and temperature regulation. So we know that, so we measure, the cold-induced VO2 max, the way we think about it is a measure of thermogenic capacity, so the ability to generate heat. And we have done experiments where we measure their body temperatures over the course of these trials too, as well. And so the ability to maintain body temperature is related to this VO2 max measure, if that's what you're asking me. I'm not sure that's what you're asking me.
I'm not sure it's what I was asking you.
One thing we do know too is that the effects of hypoxia plus cold. Well, okay, so when I showed you this, that VO2 max will improve when you acclimate them to hypoxia for both populations. The slopes of those reaction norms vary whether or not that acclimation was done under warm or cold conditions. And the lowlanders have a steeper reaction norm slope when you give them cold versus just hypoxia, whereas that slope is unaffected by cold in the highlanders. So their improvement is induced by hypoxia alone, whereas cold plus hypoxia seem to have kind of additive effects on the lowlanders. And so we don't understand that at all, and it's interesting because it's like the highlanders have very little cold acclimation capacity. Everything that's driving their plastic response is hypoxia related. Yeah.
Thanks, Zach, again for
Thank you.